In this video, I will be sharing some of the methods and key findings of our study, where we identify diseases associated with asthma using a gene regulatory network analysis. You can find this work in Frontiers in Immunology. To begin, we query Hi-C data to identify physically interacting SNP gene pairs within chromatin. We then filter for those where the SNP is also associated with regulating the expression of its target gene. Doing this for all SNPs in both lung and blood cell lines allowed us to build tissue-specific gene regulatory networks. In these GRNs, again, the SNPs both physically interact with their target genes and regulate their expression. We then located the asthma GRN within the larger lung and blood GRNs by selecting for asthma-associated SNPs and their target genes. Now, because genes do not work in isolation, we expanded up to 4 edges or levels away from the asthma GRN. Finally, we go back to the GWAS Catalog and ask which traits are associated with SNPs that are identified in each of the 4 levels. We then specifically select for the traits that are significantly enriched. What this does is it essentially allows us to find which traits are enriched in the regulatory space, which is proximal to asthma. This identified 80 traits specific to the lung GRN, which include endotypes of asthma, medications used to control asthma symptoms, and known comorbidities of asthma. Importantly, our pipeline pinpoints the genes that bridge or mediate the interaction between these identified comorbidities, meaning they can help explain the potential molecular mechanisms linking asthma to the identified trait or comorbidity. Similarly, we identified 82 traits that are specific to the blood GRN, including biomarkers and key players in the pathophysiology of asthma and comorbidities. And again, we know both the genes and variants, that are potentially mediating the interaction between them. We then wanted to exclusively identify the diseases which are observed comorbidities in the asthmatic population. And this was done by finding the diseases that are associated with our gene lists. At the same time, we used hospital records of 2 million patients in New Zealand and identified the comorbidities that are observed in the asthmatics. Selecting the diseases that are intersecting these two sets essentially identified 9 diseases. Of those, 7 had 7 diseases here, which are the ones shown beyond the red line, had higher odds of being comorbid with asthma, meaning they were more commonly observed in the asthmatic population compared to non-asthmatics. And 2 diseases were less likely to occur in asthmatics. In our paper, we also highlight which of the mediating genes are druggable to uncover new opportunities for drug repurposing. I would like to thank our funders, the Dines Family Charitable Trust, which is funding my PhD, and the Health Research Council. Thank you.